All right, today is Tuesday, May 12th for biology. So what we're going to do today is just do a few pages of reading, and then we're getting close to the end here. And so what I need you to do then is read pages 491 through 493. So 491 through 493. So this is going to be covering the activities of DNA. So the first thing we have here is the central dogma of molecular biology. That is the flow of biological information from DNA to RNA, and then from RNA to protein. It's known as the central dogma of molecular biology. So you need to know that. Reverse transcript, we have in translation, is in the bottom half of that paragraph there. It says the mRNA molecules in turn provide the information to the ribosomes to make protein molecules through translation. More recently, scientists have found that many viruses like HIV can make DNA starting from molecules of RNA in a process called reverse transcription. Before a cell divides, it makes a copy of DNA by a process called replication, which occurs during the S phase of the cell life cycle. DNA replication requires the precipitation of complex set of proteins. This process is another example of God's marvelous design in the cell. The enzyme DNA polymerase then attaches individual nucleotides to each strand of DNA molecules, forming new complementary strands. Again, it goes through the pairs. The A always goes with the T, and the G always goes with the C. Replication results in two identical DNA molecules where there had been only one. DNA has special sequences called telomeres on its end. A small part of the telomere is used during each replication. Next, we have transcription. One of the important purposes of RNA is its role as a working copy of the DNA blueprint. When a new protein is needed, the DNA coding for that protein is copied into a messenger RNA molecule. This RNA produced by transcription in the nucleus is then shipped to the cytoplasm, where it is used by the ribosomes as a template to make protein. All right, page 492. I'm going to skip down to genetic code. It took years of careful genetic experiments to understand how a sequence of nucleotides in the RNA can be translated into a sequence of amino acids in protein. Scientists discovered that the information is coded in the RNA in the form of codons. A codon is a consecutive sequence of 3 nucleotides in the RNA that translates into a specific amino acid. The equivalence of these codons to amino acids is given by the genetic code. And it gives you a table there. And it kind of describes that. I'll skip over to construction of proteins on page 493. Proteins are manufactured by ribosomes. A ribosome consists of 2 subunits, one large and one small. These subunits are normally separated in the cytoplasm but come together to make proteins. Ribosomes are made of both protein and ribosomal RNA. Amino acids brought to the ribosome by molecules of transfer RNA, which enter the ribosome through a special slot. And then we have the process of synthesizing proteins from RNA pattern is called translation, because the cell has, in a sense, translated the RNA information into a functional protein. And then we have non-coding DNA, which is one of the most fascinating features of the cell's genetic information, is the presence of large amounts of non-coding DNA, which are portions of the chromosome that do not specify the amino acid sequence of proteins. Almost all genes in eukaryotic cells include introns, non-coding DNA sequences located within the genes. Introns are transcribed into the pre-RNA molecules, but special mechanisms in the nucleus carefully splice them out of the pre-RNA, resulting in a mature RNA composed of only the coding sequences called exons. And so that's where we will stop today. No homework for today. We'll finish up tomorrow. Hope you have a good day.